So it's my pleasure to introduce Elizabeth Bradbury, who's going to talk about chasing spinal cord repair from models and mechanisms to humans. I've known Elizabeth, I guess, about 24 years. She's a professor of regenerative medicine and neuroplasticity. She's the co-director of the Wolfson Sensory Pain and Regeneration Center at King's College in London. She's also a Medical Research Council Senior Fellow. She completed her Ph.D. at the Institute of Psychiatry and her postdoctoral training at St. Thomas's Hospital and King's College in London. Her research focuses on understanding the process of injury and repair and developing therapies to restore function following CNS trauma, largely spinal cord injury, with a particular interest in glial scarring, extracellular matrix modification, immunology, and neuroplasticity after spinal cord injury. Her early mentors included Stephen McMahon, James Fawcett, and John Priestley. She's done very well in her career and received numerous awards that include the Medical Research Council Career Development Award, the Senior Non-Clinical Fellowship, the Schellenberg Prize for Research in Paraplegia, and the Suffrage Science Award, which recognizes leading female researchers. So I became acquainted with Dr. Bradbury's work through the International Spinal Research Trust meetings, which occur in London each year in the early 2000s. And she had co-authored an excellent research strategy with Matt Raymer, trying to set out the framework for a decade of research. In 2002, she published a seminal paper in Nature that showed that the enzyme chondroitinase, which is a bacterial enzyme, could promote neuroplasticity and regeneration of both motor and sensory axons after spinal cord injury. In the ensuing years, I've had the opportunity to work with Elizabeth as she advanced this chondroitinase discovery through extensive preclinical studies, including gene therapy vectors. So she's moving towards one of the first gene therapy therapeutics for spinal cord injury, and then the many steps that are necessary to move towards clinical testing, including essential development steps with the viral vector, and then with her collaborator neurosurgeon Aminul Ahmed, who actually did a postdoctoral fellowship here on a clinical trial proposal that we've been working on for the last couple of years. So she, like Mary Bunge, has diligently prepared a pathway for the realization of a career-shaping discovery into testing in people with spinal cord injury. So we're very happy to have Elizabeth present to us today.
Thank you so much. I hope you can see and hear me, and you can see my screen. I guess you'll stop me if you can't. Thank you so much, Jim. That was such a kind introduction, and actually thank you for inviting me to speak. It's such an honor to be asked to give a scientific talk at this event, remembering someone so incredible and inspirational, and inspirational to me throughout my whole career. So before I start my talk, I just wanted to say something about Mary's influence and presence throughout my whole career. So as Jim says, I've been in this field for over 25 years now. And right back from when I started as a Ph.D. student and a postdoc in the late 90s, I knew about the Bunges. They were the giants in the field. We heard their names, Richard and Mary Bunge, mentioned at every meeting, every scientific conference, and it's actually still true to this day. Sadly, I never met Richard Bunge. I never got to meet him, but Mary was this inspirational figure throughout my whole scientific career. And as a female scientist at the very top of her field, she was such a role model and an inspiration to so many of us, particularly women in our field. And I remember as a PhD student and a young postdoc wondering, would she come to my poster? Will I get to meet her at the next meeting? And then being slightly terrified if she would come to my poster. But I did meet her at a Society of Neuroscience meeting, I think way back in 1998. And she did come to my poster, and she wasn't terrifying. She was super smart. She asked really sharp questions, but she was very, very kind and generous with her ideas and her suggestions, and clearly supportive of early career scientists, and particularly female early career scientists. So, it's really such an honor to be asked to present some of my work to you people today. So yes, I'm going to talk about, well, chasing spinal cord repair. I really want to talk about going from mechanistic research all the way, hopefully, to human translation. So in my lab, we kind of have two themes. We really want to understand the mechanisms, the neurobiology of the neurotrauma of a spinal cord injury. So really understand the molecular and cellular responses that happened after an injury that lead to this chronic pathology that is non-resolving. And as Jim said, something I've been very, very interested in through my career is tissue scarring and inflammation, and particularly the extracellular matrix that's associated with tissue scarring. And we're very, very interested more in the translational side of developing advanced regenerative therapies that we hope would be translated to human. And I really want to tell a story really today about chondroitinase ABC, this enzyme therapy, and how we've really followed this story from early days to where we had a bacterial enzyme that did some phenomenal things, and we got this huge paper, to really the incremental steps and the long road to developing that, or a very advanced gene therapy approach that we really think will be something that we can take into humans to treat the injured spinal cord. And I want to show you some of our data on that in a minute. And also something we're very interested in my lab in our experimental animal models is trying to target functions that are high priority to individuals with spinal cord injury. And something I've been very focused on in the last few years is trying to recover hand function. So obviously the majority of patients have injuries at higher level, injuries at the cervical level that affect upper limbs as well as lower limbs. And so regaining use of the hands or getting fine hand dexterity back to be able to do everyday tasks that lots of us take for granted is really, really one of our big goals. And in our experimental models, we train our animals to try and do skilled reaching and retrieval tasks that really involve this fine sensory motor coordination and is very similar to the movements of humans reaching and picking up objects. So I want to go back to the kind of models we use to study in our research to try and study what's the most clinically relevant model that we can use to really understand the neurobiology of spinal cord injury and to really test our regenerative therapies to see if they really are going to work potentially in humans. So contusion, as most of you know, is the most common form of human spinal cord injury, typically as a result of a road traffic accident or falls. And after a contusion injury, so an impacting injury that bruises the spinal cord, you get this ongoing progressive tissue damage, which is called the secondary injury, which involves cell death, ongoing demyelination, pathological inflammation, and you get cavitation and scarring. And just to show you the animal model that we predominantly use, so we use the IH impactor device to deliver spinal contusion injuries. That was developed by Steve Sheff. And you can see, so these are cross sections of the adult rodent spinal cord, and you can see the intact spinal cord with the neurons and the supportive glia. And you can see even one day after one of these contusive impact injuries, you've got huge gliosis in the spinal cord and a loss of lots of the spinal neurons. But you can see that the damage evolves over time. So you've got this ongoing secondary injury that happens over the days, weeks, and even months after the initial trauma. And just to show you how similar the model that we use and many others in our field use is to human pathology. This shows a pathological sample from an individual with a spinal cord injury. And you can see this classic pathology where you get a spared rim of white matter surviving around the spinal cord. And then you've got this central cavitation surrounded by this really dense scar tissue. And it's very, very similar in the rat model, the rodent models, and the human. And I think it's really, really important to have models that reflect the anatomy and pathology of human injuries, because then we can really try to understand mechanisms of what's happening in this ongoing secondary injury. And when we have a therapy that we think is going to work, how can we need to apply it to these types of model to see if we can get repair. And it's really this area of tissue scarring. So even in spinal cord injuries that are classified as clinically complete, there is some spared tissue. You nearly always have spared axons in the white matter tracts. And actually trying to recover function in these axons, get neuroplasticity and get new connections forming is a really, really big goal of ours. And I guess there are three main targets for repair in our field, the people who are working on experimental therapies for spinal cord injury. One is to try and intervene acutely and prevent some of this secondary ongoing injury. So these will be neuroprotective type strategies. The second, which is the most challenging, is trying to regenerate the spinal cord, get back all the ascending and descending spinal connections and restore the tissue integrity. And the third, which I think is really achievable with lots of the advanced therapies that we have now, is maximizing the function, really getting some neuroplasticity, regaining function of this remaining tissue and trying to reroute pathways, rewire the spinal cord. And that really, really is achievable, I think, at this moment with some of the therapies that we have. And targeting the extracellular matrix with our enzyme therapy chondroitinase can actually have effects on all three of these targets for spinal cord injury. We've seen some regeneration. We've also seen neuroprotection. So I'll show you some of the images in a minute of our neuroprotective effects of chondroitinase. And there's a lot of evidence now that we can get neuroplasticity of intact pathways with our enzyme therapy chondroitinase. So I want to go right back to the story of chondroitinase really. Why did we ever start looking at this enzyme? And then why did we decide to try and take this enzyme to, potentially a therapy for humans? So this is based on work that was before me. So this is based on many, many years of work from people in the lab. James Fawcett and Jerry Silver were big people in this field. So what these guys showed was that these proteoglycans that are present in the extracellular matrix of the scar tissue at the injury site. So CSPGs are increased in this reactive injury scar tissue. And you can see here, this is a rodent experimental spinal cord model where the top half of the spinal cord is sectioned. And you can see this really dense reactive astrocytic scarring. And you can see nerve cells that actually grow down the spinal cord, they don't penetrate through this scar tissue. And what they showed was this scar tissue was packed full of these proteoglycan molecules. And regenerating nerve cells will go towards the CSPG molecules and towards the scar, but they never penetrate right through either the astrocytic border or the fibrotic core of the scar. So we know that now that CSPGs limit regeneration. There's also work that showed they limited transplant integration. And then it was found out that the sugar components of these proteoglycan molecules are the really potent inhibitory aspects of these molecules to growth. So that's when we started using chondroitinase. So chondroitinase is a bacterial-derived enzyme. It cleaves the glycosylated sugar chains from these proteoglycan core proteins. And there was some very nice work showing that in vitro, so cells grown in a dish, they will happily survive if you plate neurons onto sections of spinal cord or sections on scar tissue grown in a dish. But they won't put out any neurites, they won't regenerate. However, if you give chondroitinase to the culture medium, you get this beautiful kind of regenerative growth. So that stimulated a collaboration. At the time, I was a postdoc in Steve McMahon's lab. That stimulated this great collaboration with James Fawcett in Cambridge. And we wanted to know, could chondroitinase stimulate regeneration in the injured spinal cord? I was working with spinal cord injury models, mainly working on sensory systems and neurotrophic factors, but this really kind of changed my view in the field. And I started working with James closely on looking at inhibitory factors after spinal cord injury and really focusing on these extracellular matrix molecules. So this is a paper that really had, well, a big impact in my career and actually it kind of impacted some of the field into looking at these proteoglycan molecules. So we did this experiment many years ago. I mean, this was over 20 years ago now. So we had animal models that had these dorsal column crush injuries. So it wasn't the really clinically relevant models that I've just shown you. They had dorsal column crush injuries. We infused chondroitinase, and lo and behold, we got this amazing recovery. We actually were quite unexpectedly got these phenomenal results because at the time we were doing a huge combination study and we were combining chondroitinase with various trophic factors, neurotrophin-3 and chondroitinase. It was a great big study. And then we were very disappointed because lots of the animals got really sick and it turned out the neurotrophin-3 that we'd been given was contaminated. So we thought the whole experiment was a bust. We still unblinded the data because we'd just completed the study. And what we saw was that there was one group that were phenomenal. The recovery was phenomenal and it was the chondroitinase only treated group. So this really was kind of a serendipitous finding and it really changed the course of my career because then I started focusing on chondroitinase. We repeated the whole study, got the same effect. Chondroitinase treated animals recovered far better than any of the other groups. And really this kind of stimulated lots of subsequent research. There's been so many independent replications now that chondroitinase is neuroprotective and promotes regeneration after spinal cord injury. And this is from multiple labs using many different injury models. It's now been assessed in many species. There's been a lot of work in rodents, in rats and mice. There's also some beautiful work in cats by Dana Howland's lab. And these cats had a hemisection injury and they were given chondroitinase, and they recovered this really complex stepping on. They were able to step over this complex pegboard arrangement. The stepping wasn't completely natural, so it showed that there was some plasticity involved in this recovery because they found a compensatory way of walking, but they nevertheless showed recovery. Then there was a canine dog trial. So dogs with spinal cord injuries can be enrolled into clinical trials at specialized veterinary centers that do research. And these dogs had a chondroitinase injected and they recovered coordinated stepping on a treadmill. And there's now been several primate studies using chondroitinase and showing regain of skilled hand function. So really, I would say chondroitinase has the largest body of preclinical evidence than nearly all of the other experimental therapeutics that are out there. So you may think, well, this was all 20 years ago it started. Why has this not been taken forward into the clinic? So chondroitinase, there are many, many barriers to translation. One of them is the enzyme instability. It has a very short half-life. And this means you have to give chondroitinase as repeated administrations. So to go in multiple times and give multiple injections, obviously when you're thinking of translating or moving this to humans, this is not going to be ideal because you'll cause more inflammation. Chondroitinase is also a very large molecule, so there's very poor tissue penetration. And there was also limited efficacy in preclinical contusion type models. So most of the research for many, many years was in reduced models or models where there was a section of the spinal cord or a certain pathway cut. When we tried to apply chondroitinase into these more clinically relevant contusion models, it was not very effective. So this really made us think, well, we need some kind of innovative solution to give chondroitinase and give a long-term treatment and make it really penetrate the tissue and get there effectively, so it can be effective in these much more clinically relevant lesions where you've got this ongoing inflammatory pathology. So that's when we started developing a gene therapy approach, and actually a gene therapy approach can overcome all of these challenges to using chondroitinase. Before I talk about the chondroitinase gene therapy approach, I just want to highlight that there's been so many people in our field working on chondroitinase or have used chondroitinase over many, many years. And I was happy to say that Mary Bunge did have some papers that used chondroitinase, and I was very excited that she used our gene therapy for some of her Schwann cell work. And this is just a picture from one of her papers where she used chondroitinase in her transplanted Schwann cells, and actually having the chondroitinase gene there really enabled robust growth and better integration of the Schwann cell transplants. So that was really, really a highlight that even Mary got on board with the chondroitinase and saw some really effective outcomes. So to go to the gene therapy approach, so we started a fantastic collaboration with a group in Cambridge, Liz Muir, and then with Joost Verhagen, who's a viral vector expert in the Netherlands. So the trouble with it had never been possible to make lentiviral vector gene therapy of chondroitinase because chondroitinase is a bacterial derived enzyme. So it would not be able to be secreted in its active form by mammalian cells. So that meant the gene had to be genetically modified. So Liz Muir did some really clever molecular biology, did some N-glycosylated point mutations of the chondroitinase gene and made a mammalian compatible version. Then packaged this into various different vectors. He's made many different vectors over the years, lentiviral vectors, AAV vectors. And then my part of the consortium was to evaluate some of these chondroitinase gene therapies and to look at delivery efficacy in some of our preclinical models. So this has been a really great collaboration that's been ongoing for many years now, and it's really proved to be very fruitful. And just to show you the difference between the chondroitinase basic bacterial enzyme and what we see with the chondroitinase gene therapy delivery. So if you give a single injection of a bacterial chondroitinase enzyme, you can only treat a very small limited area, very localized to the injection site. And the activity of chondroitinase is not maintained. So if you see 8 weeks after this injection, the activity of chondroitinase is very, very diminished. And we have this beautiful readout to tell where chondroitinase has been active by looking at the chondroitin 4 sulfation. So wherever you've got digested stubs, so where chondroitinase is active, you reveal these digested stubs. So wherever you see green, that's where you've got active chondroitinase. And you can see with a single injection of a lentiviral vector, this is 1 microliter injected into the spinal cord. You've got this intense digestion over many segments of the spinal cord. And the digestion or the active enzyme is maintained for long term. So we've got this long-term, widespread, stable delivery with just a single administration. So I'd say the advantages of a gene therapy approach is you can get this stable long-term delivery, which I think is really important in these injuries where you've got this ongoing pathology that spreads over several segments. It's just a single administration. We don't have to go in giving repeat injections of the enzyme. And the delivery is actually very potent because chondroitinase is released very quickly from the neurons that contain the gene. So it's released in its most active form. So it's very, very potent. And because the enzyme, the chondroitinase gene is within the spinal cord, in the spinal cord cells, you don't need to worry about the tissue penetration. It's already directly in the spinal cord being secreted by the cells that express the transgene. So then we get treatments of large areas. So it's actually predominantly expressed by long projecting neurons. So it gets transported several segments up and down the spinal cord. And we've now shown efficacy in some really advanced preclinical spinal cord injury models. So just to show you the first time we applied this gene therapy approach to deliver chondroitinase in what we call our clinically relevant model of spinal cord injury and a contusion model, we did thoracic contusion injuries and we injected our chondroitinase gene either side of the contusion bruise. And you can see in animals that were untreated or if they had a control inert gene, you can see you've got this classic tissue damage at the epicenter that spreads several segments up and down the spinal cord. And then what we saw with a single injection or a single administration of our viral vector, we got this really phenomenal tissue preservation, which we were actually quite surprised about. We weren't expecting it to be this potent in terms of stopping that inflammatory pathology. But it was really, really impressive. You could see that you don't have this huge central cavitations and you've got many, many spared spinal neurons. We also saw long-term recovery of limb function with our gene therapy delivery. So with animals, what we do is we train them to cross various runways. So these animals are crossing these horizontal ladders. So they have to make a series of coordinated movements, use sensation to work out where the rungs are, and then use motor coordination to accurately cross without making foot slips. And animals with these thoracic contusion injuries, they can cross the ladder, but they make many foot slips as they cross. And you can see that animals that were treated with chondroitinase made many fewer slips as they crossed the ladder. And really nicely, this was maintained long-term through the whole experiment. And we know that that's not because they didn't have as big an injury, because we always check. The good thing about the Infinite Horizon device is you get a computer readout of the exact force and velocity of the tissue impact. So we can see that all 3 groups received the same level of injury. So this was really exciting. And we also showed improved nerve conduction with our electrophysiology in these studies. So we were really, really super excited about these results. And then we looked at how long would the chondroitinase be expressed? Is it just for a couple of months or could we see it long term? So we actually followed some animals for a whole year after injecting our viral vector. And you can see that so again, we're using this C4S staining to look where we have active chondroitinase enzyme activity. And you can see that when you inject the lentiviral vector, even 24 hours, you start to see expression of chondroitinase. It's really, really potent by 2 weeks post-injection. And you can see that chondroitinase is still active for many, many months. And we even see active enzyme for a whole year after this single administration in the spinal cord. So this is really important. Just with a single injection, we get this long-term transgene expression, active enzyme for at least a year in the mammalian spinal cord. And I think this really has wider applications, not only for spinal cord injury, but for other CNS disorders where you might want to treat large areas of the spinal cord for long periods of time, and conditions such as MS, ALS, and stroke. And we are working with several collaborators on some of these conditions. But this almost led us to a new problem. So we have chondroitinase. We know we can just do a single administration. We've got enzyme active for many, many months. But could we eventually switch the gene off? We wanted to know if we could regulate expression. Could we advance this gene therapy so that we can control it? Can we control delivery? Partly because then we'd be able to switch it off, and in terms of safety, that would be great. But also as an experimental tool to explore timings. You could then potentially switch the gene on and off at various different periods after a spinal cord injury. So again, this was a really fantastic collaboration, again with my really great collaborator, Joost Verhagen, and this work was all done by Emily Burnside, who was a PhD student in my lab at the time. So Joost made this new immune-evasive gene switch for regulating the delivery of chondroitinase. So this was a new gene therapy that you could where you could control gene expression. You could have the gene on or you could have the gene off. And this used the TET-on system, whereby you have a dual vector system, one promoter, one of the vectors expresses chondroitinase or has the promoter for chondroitinase, and one has a transactivating gene switch. And if you inject the vectors together, you give doxycycline, that activates gene expression. So this is a really great system because you can switch the gene off just by giving oral administration of this antibiotic doxycycline. So you can put the doxycycline in the animal's drinking water, and that switches the gene off, take the drinking water away, and that turns the gene off. And the other advance with this system is that Joost included a stealth gene, because the issue with using these transactivator or gene switches to switch gene on and off has always been they work very well in vitro, but when you put them in vivo in the mammalian spinal cord or in mammalian tissue, because they have a foreign body in there, this transactivator, the T cells of the CNS will destroy the cells that contain the gene. This obviously is a huge disadvantage, especially for spinal cord injury, because not only would the gene be removed, but the cells containing the gene, so it would cause more cell loss. So what Joost did, which was very clever, was to fuse this glycine-alanine repeat to the transactivator, which in effect avoids T cell recognition. So it avoids the gene being shut down and avoids you losing the cells that contain the gene. So this was a really nice study where we first assessed this novel inducible system in vivo after spinal cord injury. We wanted to see whether you could switch the gene, first of all, whether you could effectively switch the gene on and off with doxycycline. Then we wanted to see how long do you need the treatment? Could we just give it for 2 weeks and switch the gene off, or would you need it for longer? And we actually became very interested in looking at upper limb function when we were doing these studies. So we did a cervical level spinal cord injury. So first of all, we wanted to determine that we could switch the gene on and off in vivo by giving doxycycline. So animals had a spinal cord injury, they were injected with this dual vector system. Some of the animals got doxycycline daily for 2.5 weeks, and some did not get doxycycline. So one group had the gene switched on, one group had the gene switched off. And you could see that with when the chondroitinase gene was switched on, you get this really robust expression of chondroitinase gene. And that's looking at mRNA expression by PCR. And actually what we saw was expression, we saw expression of the chondroitinase transgene induced by doxycycline following spinal cord injury. And the expression was to the same magnitude as our previous gold standard vector, which was a non-inducible lentiviral vector. With the animals that had the gene delivered but no doxycycline, there was some very low level of expression. So there was some leakiness. So it's not a complete on-off gene, but it's pretty phenomenal. This is the gene off, and this is the gene on, and this is the gene off, and you can see some low-level leaky expression. Although we've never seen any biological effects of this low-level expression. But really nicely, we've shown that the chondroitinase gene, we can make a gene therapy that's inducible, so we can effectively switch the gene on in vivo after spinal cord injury and switch it off by removing doxycycline. So then Emily did this really nice work where she she really treated it like a clinical trial. She did a blinded randomized study in our experimental animals to assess short-term versus long-term chondroitinase gene on, and to look at recovery of skilled hand function. And this is following a cervical contusion injury in adult rats. So she had 4 groups. So all of the animals had a spinal cord injury. They were injected with these lentiviral, these inducible chondroitinase vectors. Some of the animals had doxycycline switched on for the whole period of time, so the long-term gene on group. Some of the animals just had short-term gene on just for 2 weeks. One group had no dox, so the gene was switched off. And then she did another really nice control, which was, well, we're giving these animals an antibiotic, maybe that by itself is going to do something. So she did another group that had just doxycycline, just to see if doxycycline on its own would have any effect. So she did a really well-controlled randomized study. Again, we showed that all the injury severities were the same, so that was nice. And then in these animals, we were interested in looking at hand function. So rats and humans have a very similar coordinated sequence of movements when they're picking up an object. So this is a really nice, this was from a really nice paper by Wishall, who's a giant in the field of hand function. And you can see that this is a rat picking up a sugar pellet and a human picking up an object. And they have this very coordinated sequence of lift, reach, and extend, then pronation, grasp, supination, and retrieval. And it's very, very similar. So it's a really nice method of looking at skilled hand function in rodents. And what we saw in these animals were that, so we had to train these animals prior to injury, and we trained them to be able to successfully retrieve these sugar pellets. And you can see after injury, they're very, very impaired on this task. There is some natural spontaneous recovery, which we always see in our experiment, in our behavioral studies, and very similar to the human situation. So there's some natural recovery that plateaus. But the only group that showed really significant recovery and which continuing recovery over time was this group that had the long-term doxycycline, so the long-term gene therapy. So that was really, really exciting. We had this new inducible gene therapy system. We saw recovery of skilled hand function following a cervical level spinal contusion injury. And it looks like you do need long-term gene expression for recovery of skilled reach and grasp. And I assume that's because we want to activate descending motor pathways for this very skilled motor task. So this was super exciting. I just want to show you some videos of some movies of these animals doing this test. So these animals so this is pre-injury. So these are adult rats that have trained to reach through this very narrow window and pick up a sugar pellet. The animals are very happy to do this task because they're greedy and they love sugar. So they'll reach and pick up sugar pellets, as many as you'll give them, basically. And it's a very skilled movement. And this animal, you can see, is very well trained. As soon as you put the sugar pellets down, it will eat it and then go for the next one. This is an animal that's had a cervical level contusion injury. So the animals we have, they're not fully paralyzed. We're really animal welfare is very important to us. So they're very mobile and they can groom, but they just don't have this very skilled hand function. So you can see the animal has some shoulder movement and some elbow extension, but it can't use those skilled digit movements to pick up the pellet. And you can see actually that the hand shaping of these animals as they're reaching for a sugar pellet is very, very similar to a human with a cervical level injury. So we really think this is a robust model for looking at deficits in hand function and for testing experimental therapies where we want to improve hand function. So this is an example of an animal that had the inducible gene therapy with the gene switched on for the full 2 months of the study. And you can see the improvements here were really quite incredible. We were so excited by this data because these animals were able to, they're not perfect, the behavior is not perfect. They sometimes, like this one, takes several attempts or doesn't get it. But the improvement compared to the no treatment or the animals that only had a short-term treatment was really, really impressive. So we were really excited by this data. We have this new inducible gene therapy system. We've shown recovery of skilled hand function. And we're currently doing work to explore the timings, the therapeutic window. We're adding some rehabilitative training, and we're working out which is the optimal vector for clinical translation to humans. So I just want to show you a few more slides of our current work. So we're really interested in trying to maximize function. So can we give this neuroplasticity promoting treatment where we get this really phenomenal recovery? But if we applied rehab as well, could we really maximize the potential for recovery? Also, it's very important when thinking about rehabilitation, it's common practice in the clinic. So it's really important to incorporate rehab and training in preclinical studies. And it's not done enough. And I think it's really, really important. And we started doing these studies. They're very challenging. We're working with Karim Fouad, who has been working on rehab and training and really been promoting the fact that you should do these studies include these rehab studies in your preclinical experimental animals. So it's really important but first for understanding the effects of neuroplasticity promoting treatments when they're combined with rehab, because potentially you're adding plasticity with your treatment and then you're adding rehab. Timing is going to be very, very important. So it might be that you want to give your rehab at the same time, or you might want to give your plasticity promoting therapy first and then apply rehab, as was shown with some of the anti-no-go work. And we think that particularly for severe and chronic spinal cord injuries, we may need to add rehabilitation to really see the maximal effects of things like chondroitinase. So in our lab, we're working with Karim, as I said, and this is a really fantastic postdoc of mine, Jared, who does these really time-consuming studies. So he's working with these cervical level injuries. He's doing these daily rehab programs. So they're really quite intense studies, but I think they're really, really important for our field, particularly when you want to think about recovering chronic spinal cord injury. So these studies are ongoing at the moment, and we're doing various other combination therapies. And then I just want to show you really the potential for this on-off gene therapy. So this is some new data from Joost Verhagen, where he's shown that you can re-induce the gene many, many times over a whole year. So this is really fantastic demonstration of this on-off inducible gene system. And potentially you can put any gene, it doesn't have to be chondroitinase. This is an inert gene, luciferase, so it means you can image in the same animal over time. You can image the gene activity. So what he's shown was that by every time you give a round of dox, you've got an increase in gene activity. And you've got doxycycline on, off, on, off, on, off. And it's really stable and consistent, even at these time points, almost a year after the administration of the gene therapy. So it's really, really important that we can show stable delivery with multiple rounds of dox. And he's also proven that this gene, the stealth gene that they've added, really does protect the neurons. So the neurons that contain the gene that had this GAR protective gene therapy are really, really healthy in the spinal cord. So this really offers many, many opportunities. You can switch the gene on and off at different post-injury time points and potentially look at windows of plasticity. You can turn the chondroitinase gene on, for example, at late stages, induce plasticity, turn it off again. And these are the studies that are ongoing at the moment. And I just want to show one other study of the chondroitinase work. And this is actually by Philippa Warren, who's currently a senior Henry Dale Fellow at King's College London and has just got tenure, which is really exciting for us. So she applied our inducible gene therapy. These animals had a C2 hemisection. They were severely impaired after spinal cord injury. They recovered to a certain plateau, and then she switched the gene on. So she switched it on 4 weeks after injury, and she saw this really nice recovery of forelimb motor score. And actually, really nicely, when she turned the gene off, the recovery was maintained. So in some models, you don't need this long-term delivery. So this is really great. It just shows another example that you can use this inducible gene therapy and apply it at late stages of injury. So I just want to really quickly go through a couple of slides to try and understand we've been trying to understand the mechanism. So chondroitinase certainly seems to be doing a lot of beneficial things. We think it's so potent in terms of repair because it targets two things after spinal cord injury. So we know there's lots of data that chondroitinase induces neuroplasticity of motor pathways, but there's also lots of increasing data that modifying the extracellular matrix really can have dramatic effects on modulating the neuroimmune response. And this really nice review article came out just as we was trying to look at these interactions, talking about these proteoglycan molecules, and they stand at the nexus of two major goals for therapies to treat CNS injuries. So increasing neuroplasticity and providing neuroprotection, so modifying that pro-inflammatory pathology. And then I really briefly want to go over some of this work where we've looked at how the extracellular matrix can modulate this neuroimmune response after spinal cord injury. So we know that one of the pathological hallmarks of spinal cord injury is the inflammation resolution failure. So there's inflammation and it doesn't resolve, unlike all the other tissues in the body. And we know that immune cell phenotype conversion is needed for inflammation resolution. And if you don't get this conversion of pro-inflammatory to pro-repair immune cells, you get these toxic macrophages at the core of the lesion, which lead to this ongoing necrosis and cavitation. And we've really shown that CSPGs actually are very important in preventing this immune cell conversion from pro-inflammatory to pro-resolution. And actually a really talented postdoc in my lab, Isaac Frankos-Kahorna, showed that we show that this is mediated by TLR4 activation. And I just want to show you how this looks anatomically, because I think it's really quite striking. So after spinal cord injury, the core of the injury is densely packed with these CSPG molecules. If you digest CSPGs, again, using our viral delivery approach, you can see that you can completely wipe out these CSPGs at the core, at the injury core. And this really has a dramatic effect on the immune phenotype at this core of the lesion. So these CD206 positive pro-repair immune cells, really are restricted. They never go into the core. They're kind of restricted to the border of the core. But when you digest these CSPGs away, you've got the center, the core of the injury is packed full of these pro-repair macrophages. So what we think these CSPGs do is prevent pro-inflammatory macrophages from turning into these pro-repair macrophages. And then if you look at longer term time points where you have these CSPGs at the core, you then get this ongoing pathology that turns into these cavities. And when you can modify the core by digesting these CSPGs, you've got this really beautiful tissue preservation. So I think this is really important that we keep looking at these matrix immune interactions and really try to understand the mechanisms deeper, because there's something really, really important here. And then just really briefly, we're moving more into these cross-translational studies where we're using our animal models and also using tissue samples to try and understand whether what we're actually working with is relevant to humans. So this was a very nice study that my PhD student Hannah McConkey is doing, and it was we have a really great collaboration with this Cynet neuron, so Sam David and Jan Schwab. So what we did, and this was when Thanos DeAngelos was in my lab, he did some proteomics in rat spinal cord injury models, identified these extracellular matrix molecules that he really believes drive inflammatory pathology. And here's a really nice example here. Biglycan, these are TLR4 activated alarmins, so they signal danger. So it's this biglycan is a proteoglycan and an alarmin. And you can see it's not normally there, but it's densely packed in spinal cord injury. And very nicely, we got hold of some tissue samples from the International Spinal Cord Injury Biobank. So they provided us with human tissue samples, a very important resource. It's very valuable tissue, but if you're looking at some new targets and you want to know if they're going to be relevant to human spinal cord injury, it's a fantastic resource, because what we saw was this biglycan that we identified in our animal models is dramatically accumulated in post-mortem spinal cord injury human tissue. So yes, we're doing lots of other cross-lab translational studies where we're really trying to use human samples and use our rat models and cross-back translate. So I just want to kind of end with where we are in our path to translation with chondroitinase. So we have this robust preclinical evidence for advancing lentichase, we're calling it lentichase therapy, to first in humans. So we've got rodent data, there's canine data, and now there's primate data. And there's really a precedent set now for regenerative therapies moving to clinical trials for spinal cord injury. A lot of this because of Mary Bunge, and of course we got the autologous swan cell transplantation and a number of other regenerative therapies that are actually currently in clinical trials. And this would have been unheard of. When I started in this field, it was completely unheard of that there would be clinical trials with a regenerative therapy, something that changes the tissue biology. This is what everyone was hoping for, but there weren't any in clinical trials when I started. So it's really, really an exciting time for regenerative therapies, and it's really exciting to be in this field. We've got a plan now to finish our advanced preclinical studies where we're working out the best rehab regime. We need to do safety tox studies. We need to get some to develop some GMP grade vectors, our IND application, MRHA approval, and then we're aiming to go for first in human clinical trials. And we have this really great team. So it's been very fortuitous that Robin Alley, who's a gene therapy expert, and he's taken gene therapies from experimental models in the eye to gene therapies that are actually being used to cure blindness. So it's really fantastic that we've recruited him to King's. He set up this gene therapy facility, and we can actually make GMP grade vectors in-house. And Aminor Ahmed, who's a great colleague and collaborator of mine, a spinal neurosurgeon, is going to be leading he's going to be the clinical lead, the neurosurgical lead for the trial. And we're working closely with Jim Guest, who's helping with our spinal cord injury trial protocol, because there's many, many years and expertise of running clinical trials for spinal cord injury. So we're super excited that that's where we are. I want to thank all these people in my lab who've done all the hard work, and all my many, many collaborators. And just if I've got a few minutes left, I know I was supposed to do a science talk, but I really just wanted to just say a few final reflections about Mary and how inspirational she's been in our field. And there's going to be many reflections on Mary later, but I just wanted to highlight one that was a really memorable for me. So one lasting memory for me is Mary's keynote talk at one of the regular spinal cord injury meetings we have, which is the International Symposium on Neural Regeneration, so the ISNR meeting. So Mary in 2015 gave a keynote lecture. It was really on her life's work, all the way from bench to bedside, culminating with the IND application. And she showed this amazing picture of the mountain of paperwork that was needed to get this IND application through. And the talk was so inspirational. So for this audience, they're all invested in this field, to see the culmination of her meticulous life's work on swan cells, and the work that was also with Richard Patwood and others. And you can see Kim and Dalton here in this picture, ending in this IND application. It was really, really inspirational. And this, Jim just sent me this picture yesterday, which is a really fun version of that. It's just really, it's such a nice picture. And she actually got a standing ovation. I've never seen anyone get a standing ovation at a science talk, but she did. It was just so inspiring and phenomenal. It was just a really nice moment, actually, and really just seemed like a really special time in our field. And actually, there's a very nice conference tradition at the ISNR where trainees swap name badges with professors. And obviously, the one that was to be really sought after that year was to get Mary Bunge's badge. And I'm very proud to say my trainee, Merrick Stratton, managed to swap badges with Mary Bunge. And that was really the highlight of his PhD. He was very, very proud of that. And he couldn't believe that she went around with his name badge for the rest of the night. And she really had fun with that. And actually, Mary was a lot of fun. And I just also wanted to just say again how important it is for us women scientists to have a great female role model in our field. And Mary certainly has been one of those, and really, really important for many of us. And I just wanted to highlight this group because there's the new generation of female scientists have set up this whiskey lean-in group. So women in spinal cord injury lean-in group, and these are the co-founders: Angela Filou, Julia Kaiser, Anna Vivanetto, and Vanessa Dulem. And really, they are the future. They're the future that are going to be leading this field and taking it forward. And they've got this amazing lean-in group for women that are working in spinal cord injury research. They have these online meetings and discuss important topics, and they support and advise each other. And I think we're very lucky in our field that we have this amazing collegiate collaborative environment, and that women feel really supported in their research in our field. And I think a lot of that is to do with Mary and her legacy, and I think it's a really important legacy to have. And of course, they also have various meetings, meetups at any conferences that involve spinal cord injury. There's a whiskey meetup. So I really advise any young trainees to get involved and join this whiskey group, because they're super supportive. And then I just wanted to, my final slide, I just wanted to end with Mary's favorite cell, beautiful Schwann cell. So these images were taken by Katalin Bartas, who was a great postdoc of mine. And she also loved the Schwann cell. And to her, Mary was obviously a very inspirational figure. So Katalin actually also gave an early career talk at the same ISNR meeting in 2015, where Mary gave her standing ovation keynote. And like my student Merrick, it then became her claim to fame that she shared a stage with the great Mary Bunge. And for a long time, I think she had the meeting program stuck up on our office wall. So yeah, she's really been an inspiration to me and to many, many other female scientists and trainees in our field. And Katalin did some great work characterizing this intriguing population of newly dividing Schwann cells they're newly dividing in the spinal cord after spinal cord injury, and they remyelinate these previously demyelinated axons. And that's a really beautiful EM image of Schwann cell. And what I really love is her description of these Schwann cells as hugging an axon. And this is a Schwann cell, and right next to an axon that it's myelinated. And this is showing that it's a similar Schwann cell that's newly dividing, and the P0 peripheral myelin that it's producing. So I just love the description of the repairing Schwann cells as hugging the axons as they provide new myelin. So I thought this would be a really, really appropriate image to end on. And then I just want to end with an enormous thank you to Mary firstly for your legacy in our field, for your work that inspired us to keep working on these regenerative therapies, and for your really pioneering studies that created the pathway for regenerative therapies to move to the clinic that many of us are now working on. It really, a lot of that was due to Mary's legacy. So thank you for really being an inspiration to all, and especially female scientists in our field. So I end there. Thank you.
Thank you, Dr. Bradbury, for that outstanding lecture. We have time for maybe one question if we have from the audience. Jim, do you have a question?
Is this on?
Yes, it is.
Okay. Hey, Elizabeth, thanks so much. That was a great tour through all that work. I wonder if you could comment a little bit on what you've done to reduce the immune response to chondroitinase, which, in its native state would be a pretty immunogenic protein.
Yes, so we have done some, so actually the modification, so I didn't have a chance to talk about that work, but the modified bacterial enzyme, so the one that's mammalian compatible, is less immunogenic. So Liz Muir, along with Pip Warren, has shown that it's, as well as being more active, it's less immunogenic. So we do think it's already less immunogenic, and we haven't, in all of the work we've done, we haven't seen a strong immunogenic response. So we don't think that it's going to be immunogenic, but obviously we need to do these safety tox studies just to be sure of that. But yeah, certainly the modified gene it does not seem to be as immunogenic as you would expect. And also we have followed these animals for long periods of time and we haven't seen any adverse events. I mean, it was almost surprising because we're giving huge bucket loads of this chondroitinase and we haven't seen any neuronal damage, we haven't seen aberrant sprouting or any pain phenotypes. So we don't think it's doing anything bad, but obviously we do need to do those formal safety tox studies and assess the immunogenicity in those formal studies.
Great. Thank you very much, Dr. Bradbury, for joining us from UK.